This video is going to summarize the other videos in the playlist which talk about blood typing. So red blood cells, they have proteins on their cell surface, not a surprise, all cells do. And a lot of these proteins are actually glycoproteins with a protein and a sugar group. So chains of sugars known as oligosaccharides are attached to these cell membrane proteins. Once again, not a surprise, that's probably true of most proteins on the cell membrane. And so if you were to ask which sugars specifically are present on these the proteins on the red blood cell membrane, there are different ones. Let's talk about two: the one that determines the ABO system and then the one that determines the RH system. The one that determines the ABO system is called the H antigen, and we can attach sugar groups. So we can attach this N-acetylgalactosamine, we can attach a galactose, we can attach an N-acetylglucosamine, another galactose, and then we can attach a fucose. If we don't attach a fucose, this is significant because now someone would, by some blood test, look like they were blood type O, but if you gave them blood type O in a transfusion, they would actually die. So this then is a structure of sugars. And if this is what your glycoprotein looks like, then you are blood type O. So what blood type O means is that you have this sequence of sugars. Now, to this base, one can attach additional sugars. So one could attach an N-acetylgalactosamine, the yellow sugar, with this yellow enzyme, if you will. And then if that's what your glycoprotein looks like, then you would be blood type A. If, however, instead of attaching this yellow N-acetylgalactosamine, your green enzyme had attached this green galactose, you would now be blood type B. So what these blood types actually mean is which glycoprotein you have. Do you have the O1, the A1, the B1, or do you have one of each if one of your parents told you to make the A glycoprotein and one told you to make the B glycoprotein? So that's what they actually look like, their sequences. But in the next animations, I'd like to simplify that. We don't have to focus on this, but I wanted you to at least see what the glycoproteins and the symbols actually represent. So here I'd like to once again simplify it. So here's a red blood cell. If someone has the O glycoprotein, I'm not going to indicate that with anything, so that will just be assumed. And so this red blood cell has the O glycoprotein. If they had attached that last A sugar, the yellow N-acetylgalactosamine, then they would have a yellow triangle, and that would represent being blood type A. So in these animations, you'll have an A glycoprotein if that's represented by a yellow triangle, or a green triangle will represent blood type B. Now, many of my students might say, well, what about positive and negative? That's coming. So let's start off with the basics, just A, B, and O. Now, this is difficult. And one of the reasons this is difficult is very often if you're studying this as a student, you're in, say, the blood chapter. And now all of a sudden here we are talking about antibodies. That's in a future chapter on the lymphatic system. And so here we're trying to incorporate a topic from a future chapter into this chapter. Sorry, no way around that. But let's just make this very general. There are white blood cells which can make antibodies, lymphocytes specifically. And antibodies will then bind to something that the body might perceive as foreign. So there can be anti-A antibodies, which would recognize the yellow triangles of A as being foreign. And then there are anti-B antibodies, which could recognize the B antibodies as being foreign. And in let's start off with blood type O. Once again, we'll add the positive and negatives subsequently. So here, let's imagine that a fetus is developing, and this fetus is blood type O. Now, this fetus has never encountered microbes in the world, so they don't know what microbes look like. and they are preparing to fight them when they encounter them someday. So they are imagining, maybe microbes will have a yellow triangle like blood type A, in which case I'll make antibodies to fight them. And maybe they'll make green triangles like blood type B, in which case I'll make antibodies to fight them. So this embryo is preparing to fight microbes of the world in the future by preparing to make anti-A antibodies and anti-B antibodies. Now, since neither of them would react against the blood cells here, because this fetus has no yellow triangles or green triangles, then there's no problem. And then this infant is born making both anti-A antibodies and anti-B antibodies, since neither react with self. So the two keys are, this individual does not have the A yellow triangles or the B green triangles on their red blood cells, and therefore they would be prepared to make anti-A and anti-B antibodies. Now let's imagine a fetus is developing and one of its parents, maybe both, said, put yellow triangles on your red blood cells by that certain combination of of sugars. Now, as this fetus develops, its immune system is preparing to fight future microbes. So here it is making anti-A and anti-B antibodies on its lymphocytes. But then at some point it realizes, wait a minute, this isn't good, because if I attack yellow triangles, I would attack myself. That would be an autoimmune disease. And so during fetal development, this individual realizes, I can't make anti-A antibodies. That would be bad. I would attack myself. So this has to go away. Right, it was a bit more dramatic. Sorry, Zoom and videos are hard to run together. And so when this infant is born, then it is born as blood type A because it has yellow triangles on its red blood cells. That's what makes it blood type A. But the only antibodies that it will be able to make are anti-B antibodies. The anti-A antibodies, the immune system realized we can't make those, so we'll have to make those cells go away to prevent an autoimmune disease. So individuals with blood type A can make anti-B antibodies, but not anti-A antibodies. Likewise, if a fetus was developing as blood type B with green triangles, at a certain point it realized, wait a minute, I can't make antibodies which would attack blood type B. I can't make anti-B antibodies, but I can make anti-A antibodies. So this individual would be born with blood type B, but making only anti-A antibodies, not anti-B antibodies, because this would attack itself. If a fetus has inherited a gene to make A glycoproteins from one parent and B glycoproteins from second parent, then it would have both yellow and green triangles. And then it would realize, wait, I can't make either of these. I can't attack myself. So if I make anti-A antibodies, that would attack myself. Anti-B antibodies would attack myself. So they would then have to go away. And so some students try to memorize this, draw a chart. So, if you're blood type this, these are the antibodies you make. That's fine, but perhaps there's an easier way. The key is you can't make antibodies against what you have. If you make antibodies against what you have, that's an autoimmune disease, you can attack yourself. So if there's an A in your blood type, you don't make anti-A antibodies. If there's a B in your blood type, you don't make B antibodies. And so feel free to make a chart that you memorize, but perhaps that's easier. You simply remember, I can't attack the glycoproteins which I have. So if there's an A in my blood type, I don't make anti-A antibodies. If there's a B in my blood type, I don't make anti-B antibodies. And so that's one system involving the glycoprotein which determines the blood type A, B, and O. There are more, however. So for example, there's more proteins on red blood cells, and you can have the KID blood group, the Duffy blood group, the MN system, the Luther blood groups. So there's a whole bunch that we can discuss. But really, the two most important clinically are the ABO system and then the system which determines the RH factor. So the RH factor is determined by another separate protein, and I'm going to use a white triangle to designate that. And so here we have another white blood cell. Now, it is helpful to consider these as being separate, the RH system as being separate from the ABO system, because it is. It's a different gene, a different white blood cell making the antibodies. And so then the question is this person A, B, or O, or AB? And that's asking the question, does it have yellow triangles, green, or both, or neither? That determines whether they're ABO. If an individual is Rh positive, that means they make the Rh factor. That means that there will be white triangles here. If they don't make the Rh factor, then they won't have white triangles, then they would be Rh negative. So this individual is O negative. They're O negative, they're O because they don't have yellow or green triangles, and they're negative because they don't have white triangles. So that's what their red blood cells look like. When we ask what are their white blood cells like, then the answer is you don't want to attack yourself. But since this individual does not have yellow, green, or white triangles on their red blood cells, it doesn't matter whether they make yellow, green, or white antibodies. So they can make anti-A, anti-B, and anti-Rh antibodies because none of these would attack self. So those are the antibodies that someone who is O negative makes. Now, someone who is O positive is blood type O because they don't have yellow or green triangles, but they do have those white triangles. They are positive, which means they have the Rh factor. And as a fetus, they realize, wait, I don't want to attack myself, so I can't make the anti-Rh antibodies, which would then attack the Rh factor. So someone who is O positive then makes anti-A and anti-B antibodies because these will not attack self. There's no yellow or white, green triangles here, but they cannot make the anti-Rh antibodies. Once again, the easiest way to remember it is you can't make antibodies against yourself. So if you have the Rh factor, the white triangles, you can't make anti-Rh antibodies. And then the remainder of this video simply then applies that for all of the other blood types. So this next individual is A negative. A, because they have yellow triangles, negative because they don't have white triangles. And then during fetal development, they realize I can't make antibodies against self, which means I can't make anti-A antibodies, which would attack the yellow triangles of self. So someone who is A negative will then make make sure that they don't make the anti-A antibodies, but they will make anti-B and anti-Rh. In contrast, someone who is A positive has both the yellow and the white triangles on their red blood cells. And then as a fetus, they realize then I can't make anti-A antibodies nor anti-Rh antibodies. So this one would attack self, that one would attack self. They both have to go. And so the only antibodies that someone who is blood type A positive would make would then be the anti-B antibodies. Someone who is B negative would be B negative because they have the B glycoprotein, the green triangles, but not the white. And so someone who's B negative can't make antibodies which attack B. So this one would have to go away and be inactivated in fetal development. But this individual could make anti-A and anti-Rh. Someone who has both green and white triangles would then be B positive. As a fetus, they realize, wait, I can't attack green, I can't attack white, but I could make the anti-A antibodies. So someone who's B positive would only make anti-A antibodies. Someone who is AB negative would have green and yellow antibodies, and they would realize, wait, I can't attack A or B, so I can't make anti-A or B antibodies, but I can make anti-RH. And then someone who would have all three, the yellow, the green, and the white triangles on their red blood cells, could not make any of those antibodies. Okay. So a blood typing question, obviously, we want to get to who can give blood to whom, has two parts. This was the first part, and actually this was the most important. So what you want to be able to determine is, given someone's blood type, what antibodies will they make? All right. And the easiest way to do this is to just remember, you can't make antibodies against self. That would be an autoimmune disease. You would attack yourself. So if you have an A in your blood type, here's the A in the blood type, here's the yellow triangles. If you have an A in your blood type, you can't make anti-A antibodies. If you have a B in your blood type because of the green triangles, you can't make anti-B antibodies. If you are positive, meaning that you have the white triangles, you can't make anti-RH antibodies. So once again, some of my students, they'll memorize a chart that they've made. The problem is during the exam, they start to get them mixed up, but it's so much simpler. You can't make antibodies against what you have. So if there's an A in your blood type, you don't make anti-A antibodies. If there's a B in your blood type, you don't make anti-B antibodies. If you're Rh positive, if there's a positive in your blood type, if you're A positive, B positive, O positive, AB positive, then you can't make anti-Rh antibodies. So once again, blood typing's it's confusing for a number of reasons, one of which is it's involving the antibodies, which you haven't studied yet. That's a future chapter. So if, this is a little hard for you, that's normal. There's nothing, wrong. It, takes a while for those because of these parts. But now we're on to the second and final step. Who can give blood to whom? All right. Now, let me warm up to this, and then I have some videos here. So let me just kind of introduce this. So here's an individual, their blood type O. Notice no yellow, green, or white triangles. And here they're making an antibody against against green triangles. Now there's different kinds of antibodies. We study that later. One of them has 5 little pieces stuck together. This specifically is IgM. Once again, we cover it later, but this is what some antibodies look like. 5 green Y-shaped antibodies stuck to a central molecule. Now, if this person who's blood type O and who makes these anti-B antibodies were then to get blood type B as a transfusion, that's bad, because look what can happen. You start to clump these cells together. These antibodies, they do clumping, and look at all the clumping that we're doing. Now, clumping is bad, and then this white blood cell, this monocyte, can come along and start to do phagocytosis, destroy all of this. So we would have a reaction against a blood type, a blood transfusion which had green triangles on it, a blood type B transfusion. In the same way, if someone who's blood type O is making anti-A antibodies, this anti-A antibody making lymphocyte can make that IgM. Once again, it has 5 stuck together. And as a result, it can clump it can clump antigens together. So if then I were to get blood type A as a transfusion, that's bad. All right, because now, all of the incoming blood can get stuck together in this clumping. Now, once again, white blood cells can come together, and let me just jump to the video. Clumping is bad. And the reason is because I have narrow blood vessels, and I don't want, clumps to block them. Destroying red blood cells, that's bad, because that then creates debris. You might say, who cares, all little cells here and there. Yeah, but once again, 99% of the cells in blood are red blood cells. So if I get, a pint of someone else's blood, I've just received, hundreds of billions of cells. If I burst them, if they clump together, that is so much debris, that is so much junk, it would block my blood vessels. And so then the risk would then be, I'm sorry, apparently we're not going there. So in this animation, here's actual blood, and I've added an antibody which will cause clumping. So let's say this is blood type A. I forget which one it was. Let's say that this is blood type A, flowing along, and I now add antibodies which stick to A. Watch what happens. You'll see, it's a little more smooth in the actual video, you'll see that it'll start to clump up to what's called a glutinate. All right, and so antibodies are going to cause that clumping that you saw. The red blood cells start sticking together. We can destroy it with white blood cells. But this is bad. I don't want this happening in my blood vessels, because now this clumping means that I'm going to block blood flow. Capillaries are narrow. And if this is going on in my circulation, and then white blood cells destroy this, this means I've just got junk, which is blocking blood flow here in my brain, here at this little spot in my heart, etc., all over my body. And this can actually result in death. So I don't want to get a transfusion with the wrong blood type. If I do, I could die. All right, because once again, there's all of this clumping that could occur, all of this debris which might result. And so then the second question then is, who can give blood to whom? So here, I'm going to say that the person receiving the blood is O negative. Now, that is so important. I'm going to repeat it a couple of times. The recipient, we're going to worry about them, and we're going to worry about the antibodies that they make. We are not going to worry about the antibodies that the donor would make. Now, I want to talk about why. One, because there's so few of the white blood cells in the blood of the donor, because they were mostly in the donor's peripheral tissues. We could then talk about the T helper cells that have to activate lymphocytes. But I don't want to answer that question because it concepts in a future chapter. And in a real class, if a student asks this question and I answer it, it clarifies the situation for some of the students, but then some of the students say, oh, I understood it until a minute ago, and now I have no idea what's going on. So I'm not going to try to explain why in great detail. So once again, we're going to ask what antibodies are made in the recipient, not in the donor. So if the recipient, the person getting the blood, is O negative, we ask what antibodies the recipient makes. This is what we just talked about, all right? So you ask, all right, if someone's O negative, all right, they don't have yellow triangles, they don't have white triangles, they don't have green triangles. So they will make antibodies against all of these because none of these will react against self. So if I'm O negative, I make anti-A antibodies, anti-B antibodies, and anti-RH antibodies. So that was step one. There's two steps in solving this problem. Step one is, what antibodies will someone make? In this case, the recipient. Once you know that, you've then answered your question. You now know the blood this person can get. If I attack A, you can't give me blood with an A in it. If I attack B, you can't give me blood with a B in it. If I attack the RH factor, the positive, you can't give me blood which is positive. So once you know what antibodies someone makes, you now know the blood that they can't receive. Because if these are the donors, so here's donor blood, which is A positive, A negative, AB positive, AB negative, B positive, B negative, O positive, O negative. Here's all the possible donors. Antibodies will stick to all of them except one. So if I'm making anti-A antibodies, I will clump and attack blood with an A in it. If I'm making anti-B antibodies, I will clump and attack blood with a B in it. If I'm attacking the Rh factor, even blood which is O positive, which lacks the Rh, which lacks A and B, has the Rh factor, so I will attack that. The only blood type that I will not reject is O negative, because I'm making anti-A, anti-B, and anti-Rh antibodies. There's only one blood type which has none of those. So if I am O negative, I can only receive O negative blood. I will attack all of the others, all right? And once again, you know that by saying, all right, the recipient, the O negative recipient, what antibodies will they make? And then very simple, that's the blood you can't give them. Now, if the recipient was O positive instead, now the recipient is O positive, so that's what I say here. They have white triangles on their red blood cells, so they can't make the antibodies which would attack the white triangles. So an O positive individual cannot make anti-Rh antibodies. Once again, I apologize, I'm just slowing this down. But they can make anti-A and anti-B. And so once you know that the recipient will make the anti-A, attacking the yellow triangles, the anti-B, which attack the B anti-B triangles, now you know what to do. You can't give your patient, blood with an A in it because they attack A. You can't give your patient blood with a B in it because they attack B. So you ask, what antibodies will this O positive recipient make? They make anti-A and anti-Rh. And now you know I cannot give them blood with an A in it. I can't give them blood with a B in it. But if it doesn't have an A or a B, I can give them the remaining blood types. And so here you see that an O positive recipient can receive both O positive and O negative blood. Now, once again, some students, they try to make a chart, they try to reason through it. Wait, can the O positive receive O negative? But the easy way is, once again, you ask, what antibodies does the O positive individual make? They make anti-A, they make anti-B. Notice that if you don't make anti-Rh antibodies, you don't care. You don't care if the blood from a transfusion is O positive. You don't care if the blood from the transfusion is an O negative. You won't attack it either way. You don't attack the Rh factor. You don't attack it if it's present, and you certainly wouldn't attack it if it's absent. So then this individual can receive both O positive and O negative. And then remaining the videos then go through blood types, A, B, and then the transfusions. And so here they are separate, and feel free to then play these as many times as you like. So if the recipient is A positive, once again, that's the recipient, you ask, what antibodies would a recipient who's A positive make? And the answer is obvious. They can't make anti-A, because that would mean they would attack themselves. They can't make anti-Rh because they're A positive. They have the Rh factor. So all they would make is anti-B. So a recipient who is A positive would only make anti-B antibodies. And now you know what you can't do. You can't give them a blood transfusion from a donor with a B in it. They attack blood with a B in it, so you can't give them B positive, B negative, AB positive, AB negative. But anything that doesn't have a B is fine. If they are A negative, not only do they make anti-B antibodies, but they would also then make anti-Rh antibodies. So they don't make anti-A. If you're blood type A, you don't want to attack yourself. But an A negative recipient would make both anti-B antibodies here in green and anti-Rh antibodies in white. So you can't give them blood with a B in it, and you can't give them blood which is Rh positive. So nothing which is positive, that would include A positive and O positive, and nothing with a B in it. So this person could only receive A negative and O negative. Once again, some students make a chart. Okay, so a recipient who's A positive can get A positive and A negative, O positive and O negative. A recipient who is A negative can only get A negative and O negative, and they make a chart. But once again, during an exam, memorizing that chart, they could all rush together. And so I think it's just easier. First, you ask the recipient, what antibodies do they make? And then simple, don't give them blood, that those antibodies would attack. If they make anti-A antibodies, you don't give them blood with an A in it. If they make anti-B antibodies, you don't give them blood with a B in it. If they make anti-RH antibodies, you don't give them blood that is Rh positive. And then, that I think is the easier way. And the same thing here, B positive individuals make anti-A antibodies. So don't give them blood with an A in it. They'll attack those cells. All of that clumping, all of the debris could potentially block blood vessels. They could die. Okay. Now, these videos go through all of the scenarios. And before we end, there's just something that's annoying that we have to answer. And I know you will be annoyed by this, and sorry, there's no way around this. So, if this patient was B negative, they make anti-A and anti-RH antibodies. You can't give them blood with an A in it. And if you are then asked why, the answer is because they could die. All right? Because the clumping and the destruction and all the debris could clog blood vessel here and there and there and there, and they could die. So if you are asked, what would happen if you give blood type A to someone who makes anti-A antibodies? You would say, that's bad, they could die. If you're asked, what would happen if I give blood type B from a donor to someone who makes anti-B antibodies? You would say, that's bad, they could die. So some transfusions you need to avoid, your patient could die. Some would be fine. So here in this case, this B negative individual could accept B negative blood and O negative blood because they're only attacking A and the Rh factor. These two blood types do not have anything that this recipient will attack. And so if you're not making anything that this recipient will attack, then this is a fine transfusion. What would happen if we give this B negative individual B negative blood? Oh, that's fine. What would happen if we give them O negative blood? Oh, that's fine. All right. So some blood transfusions are fine. However, here's the tricky part, and I'm sorry, you're going to hate this at first, but it'll ultimately make sense. What is the best answer to say? What would happen if we gave this B negative recipient O positive blood? The best answer is the first time it might be okay, and then after that it would be life-threatening. Okay, let me repeat that. If the recipient is Rh negative, this only applies to Rh negative recipients. What would happen? The best answer is the first time it might be okay, but the second time it's potentially life-threatening. And you're confused, obviously, and you ask, wait, doesn't this B negative individual make anti-Rh antibodies? So if you gave them, say, O positive blood, wouldn't they attack it and potentially die? And the answer is the second time, yes, but the first time, maybe no. Let me explain, and this is then our final concept. If you're confused by this, there's a good reason, because this involves the immune system and the humoral response where we make antibodies, and we haven't gotten there yet. So let's just discuss this. So here we have blood type O negative, A negative, B negative, AB negative, and here we have blood type O positive, A positive, B positive. And the difference is the Rh factor. All right, so some of these have the white Rh factor if you're positive, and then you're negative. So O positive has the white Rh factor, O negative doesn't. Both A's have the yellow triangles, but A positive has the yellow Rh, my fault, has the white Rh factor, but not A negative, etc. So the thing that makes A positive different from A negative is the white Rh factor. What makes the B positive different from the B negative is the white Rh factor. AB positive from AB negative, the white Rh factor. So the positive people do not make antibodies which will attack Rh, because all of the positive people have the white Rh factor. So none of these positive people want to make antibodies which attack Rh, because they'll attack themselves. So if you're positive, you don't make antibodies against the Rh factor. That would attack yourself. But if you're negative, O negative, A negative, that's O negative up there, O negative, A negative, B negative, AB negative, you do then make antibodies which would attack the Rh factor, which you don't have. All right. So the people who are negative, they can't receive a transfusion from a positive source because they will attack it with these antibodies. But the thing about antibodies is we don't really make them after our first exposure. In our first exposure, your body is learning what will be something that's foreign that needs to be attacked. So we make antibodies after our second on our second exposure in high quantities, and that's an important concept in the immune system chapter. Now, I use this very silly analogy in my classes where I say, if someone with a green mohawk, or like say a green mohawk gang, robs a bank in your town, were the police likely to stop it? And the answer is no. They're not looking for a gang with green mohawks. They were taken by surprise. But once they know that the green mohawk gang robbed this bank, they're now looking for the green mohawk gang. And now they're much more likely to stop the next green mohawk gang bank robbery because they're saying, aha, we are going to double our surveillance. We're looking for the green mohawk gang. In the same way, if these individuals got a transfusion from someone who was Rh positive, they weren't looking for that the first time. They weren't making all of these white antibodies against Rh positive the first time. They were taken by surprise. But once they got that first exposure, then in the future, they would make large quantities of these anti-Rh antibodies. So then the second time, you would see the clumping, the immune reaction. So after the second time, it would be potentially life-threatening. So technically, if you asked what would happen if A positive donated to A negative, the best answer is it might be okay the first time, but after that, a problem. What would happen if B positive donated to a recipient who was B negative? Might be okay the first time, but after that, it's potentially a problem. So technically, that's the right answer. We make this huge antibody response after the second exposure. The first exposure only sensitizes us to this. Now, what I'm saying is true when the recipient is negative. So that's true of recipient who's A negative. What happens when you give them A positive blood? Okay, the first time, the problem but you can't do it the second time. That's not true of this category. If the recipients are positive, they don't care. So O positive can receive O positive and O negative blood. A positive can receive A positive and A negative blood. They don't care. If you're a recipient that is positive, you don't make anti-Rh antibodies. You don't care if the incoming blood is positive or negative. So everything that I've just said applies to the recipients who are Rh negative. Now, there's two scenarios. One is you're asked, say on an exam, what would happen if an A negative recipient receives A positive blood? The most correct answer is that it might be okay the first time, but after that, potentially life-threatening. And you're confused, and I get it. You might have to watch this video again, or the individual videos that just go through the Rh factor. And not only are you confused because you haven't studied the immune system yet, but you're annoyed because you're thinking, really? Are blood banks that incompetent that they're constantly messing up? And we often say, phew, at least this was the first time you gave Rh positive blood to an Rh negative recipient. If it was the second time, then it would have been a problem. No, the reason that this is such a big deal is because of pregnancy. So this is the last topic in that. You may have noticed that the white antibodies in the previous scenario, They were single, like a single Y-shaped structure, not that group of 5 that you saw before. All right. Now, it turns out there's different kinds of antibodies. IgM, that comes in the group of 5. IgG, which can be single. IgG can cross the placenta because it's smaller. IgM, the group of 5, cannot. So now let's consider 2 scenarios. Let's say a mother is Rh positive, but her fetus is Rh negative. No one cares. The reason they don't care is because if you are Rh positive, like this woman is, you don't make antibodies against the Rh factor. So this mother's immune system doesn't care. If this fetus is Rh positive, she won't attack it. If it's Rh negative, she won't attack it. So she doesn't make anti-Rh antibodies. She doesn't care. So everything that I'm about to say does not apply to mothers who are Rh positive. She will tolerate both Rh negative and Rh positive fetuses. But the scenario is different if the mother then is Rh negative. Rh negative women can make anti-Rh antibodies. So if the father was Rh positive and the fetus is Rh positive, this is then potentially a problem because this woman would recognize white triangles in fetal blood as foreign, but not the first time. Because once again, with that green Mohawk bank robber, could the police department in your town, go after a green Mohawk gang? Yes. But until the green Mohawk gang has done the first bank robbery, the police aren't looking for the green Mohawk gang. So no, they're not going to go after them the first time. Same thing here. If this fetus then has white triangles because the father was Rh positive, this fetus is Rh positive. Could the mother make anti-Rh antibodies which cross the placenta and attack the fetus? Yes, she could. But if the mother has never been exposed to Rh positive blood, she's not before that first exposure. So this first pregnancy occurs normally. But during childbirth, blood is exchanged, the placenta tears, and now the mother's, sorry about that. During childbirth, the placenta tears and the mother's immune system is exposed to blood with white triangles for the first time. And then the mother's immune system says, oh, look at these white triangles. We've never seen them before, this foreign substance. We'll be ready the next time. So after this first encounter, then the mother's immune system realizes we're going to make antibodies against white triangles from now on at a higher level. If this woman becomes pregnant another time with an Rh positive fetus, then the antibodies that her immune system is making, because they are IgG, the smaller ones, they can cross the placenta and now destroy the white blood cells in this fetus, causing a number of potential negative effects, including death. So this Rh negative mother's immune system can attack the white blood cells from Rh positive fetuses, not the first time, but after subsequent births. So I'm old, and when I was a child, I'm Rh positive. And I was fine because I'm the first in my family. But then there was the problem is, would my mother's immune system potentially attack the blood cells of my younger siblings? That was an issue. However, a substance was developed called Rogam. Rogam in people younger than me and my family, Rogam clumps fetal blood. So it's given to Rh negative women. So if a mother, a pregnant woman is Rh negative, she is then given Rogam. Rogam will now clump fetal blood with the white triangles and prevent this first encounter. So remember, what caused the encounter, And what caused this is that the mother's immune system touched white triangles. But if you can clump that together first, get to it before the mother's immune system, you prevent this encounter, and you prevent then the mother from making antibodies against the Rh factor from that point on. So Rh negative women are then given a substance ROGAM, which will prevent this clumping. And so therefore, there is no longer as great a concern with Rh negative mothers. Now, occasionally there still is a problem. So for example, did an Rh negative mother maybe experience a miscarriage? In which case, before ROGAM was administered, and then she did develop those antibodies, then that is a concern. But for the most part, after, say my generation, ROGAM became standard to give to Rh negative pregnant women. And so therefore, this is no longer an issue. Finally, forgive me, I teach genetics. And so I think this is fascinating, but, so let me just end here. If you take any species, let's not consider humans for a second, and they spread over a big planet, and you have a population over here and a population over there, and they vary over time. So with dogs, the dogs from this continent look different from the breed of dogs from this continent. After a certain amount of time passes, the genetic differences can accumulate so that these two breeds or these two populations are so different that if they then try to interbreed, it doesn't work well. And the immune systems of females can then attack, fetuses, from the other, and you can cause a whole host of issues. After a certain point, these two breeds, they really can't interbreed well together. The embryos don't tend to make it to birth. They're sickly afterwards. There's a whole host of issues. So that happens in any species after a certain degree of genetic difference. So it's a legitimate genetic question. You can ask, well, human ancestors, we all come from the same ancestors. Your great-great-grand-somethings from my great-great-grand-somethings. But as they spread to different continents and they varied over time, you could ask, it's a legitimate genetic question. Did any two human populations get to the point where now interbreeding should probably be discouraged because they are genetically distinct so that their fetuses would not thrive because of a variety of genetic differences? And the answer is a resounding no. That even though there's all of these different human populations, at no point did genetic differences reach the point where breeding between human groups is a problem, with one exception, and this is it. Because imagine a scenario where you have a mountain range and the people on one side of the mountain range are Rh negative, people on the other side of the mountain range are Rh positive. You build a tunnel between the two mountains, and these groups, they can intermarry and interbreed, but the Rh negative women will attack the second and third and fourth fetuses fathered by Rh positive fathers. And so here's a scenario where there would be a potential separation between these, where they're almost better off reproducing among themselves and not trying to interbreed. Now, there have been different species of humans in the past cohabitating. So, about 100,000 years ago, even 50,000 years ago, there was more than one species of the genus Homo. So that's certainly possible that there are different species. This is the only example that we know of where our one human species might actually have been taking the first tentative steps into splitting into two because of this incompatibility. But because of ROGAM in the modern world, no longer an issue, so no longer a factor, but still just genetically interesting that we are one species and there is no problem in intercrossing between population on this continent from that continent, with this one exception, which once again ROGAM addresses. So that was an overview of blood typing. All of my videos go through the different steps in a different order. And what I would recommend to my students is you solve a blood typing question in two steps. One, you ask the recipient, what antibodies do they make? And that's easy. Don't memorize a chart. Just remember, you can't make antibodies against what you have. So if you have an A in your blood type, you don't make antibodies. If you have a B in your blood type, you don't make anti-B antibodies. If you're positive, then you don't make antibodies against the Rh factor. Second step, who can give blood to whom? And then you ask, all right, well, let's look at the recipient, not the donor, the recipient, and ask what antibodies do they make? If the recipient makes anti-A antibodies, then don't give them blood with an A in it, they could die. Does the recipient make anti-B antibodies? If they do, don't give them blood with a B in it, they could die. If there's no reaction, then if there's no antibody in the recipient, which will act on the donor blood, then that transfusion is fine. So for some transfusions, you say, no, don't do it, they could die. On other transfusions, you say, oh, that would be fine. Then finally, there's this third category. If the recipient is Rh negative, this only applies when the person getting the blood is Rh negative. What I'm about to say would never apply to someone who is Rh positive getting blood. If the recipient is Rh negative, if there's no problem with the AB system, but the incoming blood is positive, so if I'm O negative and I'm getting O positive blood, what might happen? The first time it might be okay, but then the second time after I've been exposed, I'm making antibodies, then there would be a potential reaction. So that's a summary of blood typing and the blood typing problems. Once again, all of these videos go through it a little more slowly in individuals, so feel free to visit those individual videos to go through anything that you would like to review again.